Welcome, Eastview Scholars, to this edition of Sharp Shorts. Let's go to another world and talk about transport. Obviously, we are multicellular organisms that one of the things that makes us a living organism is that ability to separate our environments for our health and safety. Now, we do so with the selectively permeable membrane in our cells, which keeps the extracellular fluid away from the intracellular. And you can see here, being selectively permeable allows you to control, and we have talked about the plasma membrane and its role, but it just allows you to control what comes in and out to keep that homeostatic balance in your body. Okay, what are some of those things that balance it? The size of the molecule, the charge of the molecule, maybe its shape. Is it lipid soluble? Is it gas soluble? Other factors like that. And the role of proteins and stuff in the structure of the plasma membrane and allowing things to come in. Okay. Two types of transport, passive and active. Active requires ATP. Your body does much more with passive diffusion, osmosis. Facilitated diffusion or carrier-mediated transport is another way to say it. Much more passive because you just don't have the ability to expend that much ATP for all the things that are flowing in and out of your cells all the time, and the work that would require. Okay, diffusion. Most of you know what that is. That is just, here's a very simple example, but diffusion is just moving from, as all passive transport is, going from a higher concentration to one of lower. May or may not be evenly distributed, all depends, but here you can see a sugar cube that dissolves. Sugar cube with dye in it. Anyhow. In extracellular fluid outside the cell, the waters and the solutes can diffuse freely. They are selected against the plasma membrane, and for us, things that could move across are lipids, lipid-soluble molecules, soluble gases. Here, we'll use this picture here. You can see here, lipid-soluble molecules and gases can diffuse across. You can get some leaking of very small ions and stuff like that. Small water-soluble molecules and ions can pass through membrane channels. Some of those may be controlled, as for sodium and potassium reactions and stuff like that. Large molecules cannot fit, it cannot diffuse, and they can only carry, there can be facilitated diffusion, there can be active transport, there can be stuff going on to allow those. Okay, and again, factors that influence the rate that those things can be is the distance that they have to travel, the size of the molecule, maybe what the temperature is for the organism, or the size of the gradient, the electrical forces, osmosis, and again we'll go to our water picture here. Osmosis is when the solutes themselves cannot pass through the permeable membrane. Okay, so right now we have an equal water volume, but we do not have an equal molarity. What happens then is the water will diffuse to even out the molarity, although then the water volumes will be different. And so that flow of the water is, movement of the water is driven by osmosis, and that can create a pressure. Okay. Hydrostatic pressure is if we were to put a pressure on the force to stop and balance the osmotic movement, then that would be equal to the osmotic pressure as you balance them out. Okay. The osmolarity, the concentration, is very important in certain things. You know, not everything needs to be a gradient. That is great for movement. And let's go to the next. This is a great example of why you need to be careful of movement and controlling movement and stuff. Here you have a hypertonic solution. So as you can see, the extracellular fluid would have many more solutes than inside the red blood cell, and the water escapes. And what you get are called crenellated and not very useful red blood cells. Here, the balance is equal as it should be. You're isotonic. You have the same molarity concentration inside out, and that keeps your red blood cells happy, and the water, is in balance. Here's hypotonic. You can see this little guy has burst or lysed, and that's because now that inside your cells, many more solutes, the molarity is higher than outside, so you get water flowing in from the extracellular fluid, and that eventually can cause red blood cells to burst. And that's not the only thing. We use red blood cells as an example, but then it gives you an idea for every cell how things move in and out. And so carrier-mediated transport, things that are hydrophilic or large molecules may need help either through co-transport or counter-transport or exchange pump to get through. Okay. And that would be facilitated diffusion, which requires no ATP. That would be active transport, like sodium-potassium pumps, which we will look at in more detail in class, or the secondary active transport. The mechanism doesn't require, but the cell might need the ATP to maintain homeostasis. Where you'd see an example of this is resetting the sodium-potassium balance after stuff, so the active transport goes to balance things out. All right, and here you can see, here's an example of facilitated diffusion, where you need the protein that specifically helps bring in that glucose molecule in that carrier protein. And when it binds there, it conformationally changes that protein. It opens up and allows that glucose molecule in. Here's our sodium-potassium pump. This is electrically controlled, and much more complex than we're going to talk about in this video. We'll discuss it more in class. But essentially what you do is you have some stimulus, but you open up this gradient and you can get the flow of sodium and potassium, but then you have to reset the gradient, and that requires some active transport, of which you pump out 3 sodium and 2 potassium in. Okay. Secondary active transport, you can see this is one of those things that happens when you get the rush in, you get that differential, and the sodium ions want to come flowing in, and the glucose molecule takes a ride. But again, to reset the balance in the pump, you need to pump the sodium out. Okay. Last thing we'll talk about in the cells is how do they get their food or their water and stuff in and out, and how do those vesicles do. Vesicles are formed as things that pinch off. You have either endocytosis or exocytosis. It's really the same thing. But bringing in food or water or things along that line, you can see that there is a trigger at the plasma membrane to, say, surround a vesicle and bring in the stuff that you need and pinch it off. Then, of course, our friend the lysosome comes in, say this was food products, would come in and help digest the food products, and then you could go back. You could do the same process the other way, that you had stuff that you wanted to get rid of, say some cell waste or something like that. You could have the fusion here, and then the lysosome would bind. Maybe you made a vesicle internally surrounding some waste product, some protein waste product or something along that line, or a damaged mitochondria. The lysosome would bind, digest it, and then you could go that way and use exocytosis to get rid of the waste. Okay. So, endocytosis again brings in water and cell. Exocytosis will discharge materials that are have been digested in our waste products. Okay. Here's an example of pinocytosis. You can see it pinching off and bringing water into the cell, so the cell can enjoy that. Here you can see a little exocytosis. You surround a bacterium, the lysosome comes in, digests it, and by exocytosis. So there's a quick look at transport with your book and our classroom activities. Hopefully that gives you a full understanding. Anyhow, Scholar on, Eastview.